Today our grand round will be by Dr. Altaf. Dr. Altaf is well known in our genetic lab. He is PhD from India, training in United States, and he worked as associate professor and the head of ophthalmology genetics lab in Department of Ophthalmology, King Saud University. He is associate with glaucoma research chair, and his area of research is focused mainly on glaucoma. Today he will present a topic about genetics of eye disease. Welcome, Dr. Altaf.
Assalamu alaikum and good afternoon, everyone. Thank you, Professor Sam, for your kind words and for agreeing to moderate this grand round. So today I'll just present my talk on genetics of eye diseases. It's just kind of an overview. So the topics that we'll be talking about is basic genetics with genetics of eye diseases and their mode of inheritance with examples. And then lastly, we'll talk something about diagnosis and management options that are available as of now. So just as a quick overlook at the basic genetics, I'm sure all in this room must be aware what are genes. Genes are basic hereditary and fundamental units of life. They are located on chromosomes, which are made of DNA and proteins. And genes contain instructions that make proteins which carry out a wide range of functions. What are the basic functions? They code for proteins that support bodily development, physiological functions, and many of our physiological traits such as eye color, hair color, and height. They also play a crucial role in both our well-being as well as disease development. Besides, they can cause disease as the result of mutations and alterations in the DNA sequence. They can result in defective proteins and alter structure and functions of protein, either by causing loss or absence of proteins. And as it is said that genetics actually loads the gun and environmental factors pull the trigger, so they can very well interact with environmental factors to increase the risk of developing a disease. So what's a genetic disease? Can be defined as a health problem that arises due to one or more abnormalities in the human genome. Importantly, it can be passed from onto an offspring from one of both the parents or occur spontaneously. What are the causes of genetic disease? There are mutations, which are changes in the DNA sequence. There are gene deletions, where there is a loss of a portion of DNA. Gene duplication, which is a gain or of a copy of portion of DNA. And chromosomal rearrangements, which is changes in the structure of the chromosomes. And what are the different types or categories of genetic disease? There are monogenic disorders, which are caused by mutations in a single gene. Then there are polygenic disorders that are caused by mutations, and the name suggests in multiple genes. Then there is complex disorders, which is caused by combination of genes and alterations in the environmental factors. And then there are chromosomal disorders, which is caused by damage in the chromosomes. What are the molecular mechanisms? As I mentioned earlier, there are mutations that occur, so that can be a gain of function mutations, where mutations can lead to gain of function effect, causing proteins to be overactive and exhibit abnormal functions. This can lead to cellular imbalances and contribute to disease pathogenesis. In contrast, there can be loss of function mutations. Loss of function can lead to production of non-functional or truncated proteins, resulting in deficiency of the protein's normal functions. This can also disrupt many cellular processes and contribute to disease development. Besides that, there can be disruption of protein structure. These mutations can cause amino acid sequence changes, which can cause altered protein conformation and impaired functions. These can affect various cellular processes, including protein folding, enzyme activity, and signal transduction. There can be defective protein interactions. Due to mutations, the protein and protein interactions can be disrupted. This can result in formation of complexes that are unable to carry their function. They can disrupt signaling pathways, cellular trafficking, and other critical cellular processes. So moving on to the genetics of eye diseases, and we'll see what are the mode of inheritance. So what can be defined as a genetics of eye disease? Eye disease resulting from inherited genetic defects are referred to as genetic eye diseases or inherited ocular diseases. Over 500 genes have been reported so far that contribute to various inherited disorders, including retinal dystrophies, cataract, glaucoma, age-related macular degeneration, and many others. Due to advances in next-generation sequencing, genetic testing is now becoming increasingly important in ophthalmology, and it can be used to diagnose inherited eye diseases, identify individual risk of developing the disease, and guide the treatment and management decisions. So we'll have a look at what are the pattern of inheritance with some examples of eye disease. So inheritance pattern describe how genetic traits are passed from parents to offspring. Inherited or genetic eye disease can follow various patterns of inheritance: autosomal dominant, autosomal recessive, X-linked, mitochondrial, or they can be polygenic or multifactorial. So let's have a look at what is autosomal dominant inheritance. So this is a cartoon over here which shows how the inheritance patterns takes place. So as the name suggests, autosomal means the mutation is located in the first 22 pair of the chromosomes. They are located in the autosomes and not in the sex chromosomes. And dominant means that only a single copy of mutated gene is enough to cause the disease. Now if you can see in this cartoon, there is an affected father who is having only one pair of one of the pair is affected, and still he is getting the condition of the disease. And what are the chances of such parents passing the offspring, passing the gene to the offspring? So affected individuals have 50% chance of passing the mutated gene to the offspring. You can see here, like, out of the 4, you have possible of 2 of them having the disease. So an example of autosomal dominant eye disease is aniridia. So there are many examples. I've just selected certain of the things, so it's not that only this is the one which is autosomal dominant, or whatever examples I will use. So aniridia is a rare genetic disorder which is caused by eye malformation characterized by partial or a complete absence of iris. So aniridia is the complete absence of the iris, and this is an autosomal dominant inheritance, and most of the mutations which caused by autosomal dominant account for 2/3 of aniridia cases. It is primarily caused. Okay. So it's primarily caused by mutations in PAX6 gene on chromosome 11 or deletion of this regulatory regions that control PAX6 expression. So this is just its gene organization. Thank you. So this is the gene organization of PAX6. Which is the exon intron organization, and these are the different protein isoforms. As you can see here, they vary they have 2 isoforms here, which is just depending on the presence or absence of this 5A exon. Anyways, so this gene, PAX6 gene, has 14 exons, and it codes for around 422 amino acids. It's a transcriptional regulatory protein, and the protein plays an important role in eye development, neural tube development, and olfactory buds and pancreatic tissue development. This is the protein structure of the PAX6 domains, different domains based on the exon organization. And there are nearly 300, more than 300 PAX6 mutations have been identified. Most of them cause aniridia and are also associated with congenital eye malformation, or cause related ocular phenotypes such as Peters anomaly, foveal hypoplasia, and optic nerve abnormalities. Coming to the next one, which is autosomal recessive inheritance. So what is autosomal recessive? In this disorder, there are 2 copies, as in contrast to autosomal dominant 1, which requires only 1 copy. Recessive requires 2 copies of mutated genes to be expressed with the trait or the conditions. As you can see in this cartoon, that both the parents are harboring one of the abnormal gene from the one pair. And person with only 1 mutated gene will not have the trait, but they can pass the mutated gene to the offspring. So you can see here they are not called affected, they are called carriers. If you carry only 1 of the abnormal gene, they are referred to as carriers. But if both the parents are carriers, they have a 25% chance of having an unaffected child or an affected child. So you can see here out of 4, you have 25% chance of having an unaffected child, or 25% chance of having a affected child, and about a 50% chance of having a carrier. So an example of this disease is achromatopsia, which is referring to color blindness. It's a rare bilateral recessively inherited disorder affecting cone photoreceptor cells, causing reduced visual acuity, photophobia, hemeropia, and severe loss of color discrimination. So what it shows in the cartoon is like the photon light activates opsin, which is then activates the transduction protein called GNAT2, which in turn activates phosphodiesterase PD6 that hydrolyzes the cyclic GMP to GMP conversion, and cyclic GMP keeps the cyclic nucleotide channel. This channel is open, it is regulated by this conversion. So eventually then the color signal is transmitted to the brain. So that's how a normal pathway of color vision looks like. And it has been seen that genes that code for this cone cyclic nucleotide gated cation channel, CNG channel subunits, are most commonly affected in this disorder. This includes the CNGB3, which accounts for 50% of the cases, and CNGA3. So what I'm talking about is these channels. The mutations in these channels, they contribute for most of the cases. And approximately over 100 mutations have been reported so far. And there are also smaller fractions of this chromatopsia, which is caused by mutations in GNAT2 or phosphodiesterase esterase genes and ATF6 genes, which causes a truncated protein, a non-functional protein, which can hinder that photon effect of cyclic GMP levels, thereby blocking the transmission of color signals. Now moving to the next one, which is X-linked inheritance. Now X-linked can be recessive as well as dominant. So what happens, as I said, in recessive, you will only you'd require both the copies to be present in the pair. But in this case, you can see since it's X-linked and the father is having only one chromosome which is abnormal, so it is still affected. That means because they have another one which is Y, so you actually have one X gene. That's why you express this trait. So in this case, affected father where the abnormal mutated gene is on the X chromosome and the mother is unaffected. So father with such a disorder will pass his X chromosome to his daughters, who will be carriers, and sons will be unaffected. So in this case, you can see the father is passing the gene X, the abnormal X gene to the daughter, but she's receiving a normal X gene from the mother, and that's why she's a carrier and she's not affected. And in this case, what happens is the son will always remain unaffected because they are never going to receive the X chromosome from the father. That's why they will always remain unaffected, and the daughters will have a chance of being a carrier. And in case when the scenario changes where the mother is a carrier, like she has two pair of X chromosomes, but only one of them is affected now, so she's a carrier, unlike the father who was affected. So what happens in this case is each daughter will have a 50% chance of being a carrier. Likewise, each son will have 50% chance of inheriting the abnormal chromosome and being affected. So there is So there is a chance that mother can pass the abnormal chromosome to the son, so he can be an affected child, whereas the daughters will be carrier or unaffected. These are the number of diseases that are linked to X chromosome: ocular albinism, cone dystrophy, Norrie, and all these things. These are all X-linked disorders. Not necessarily they're all recessive, but these are all of them. Some of them are dominant also in that. So X-linked disorder is an example is Norrie disease. Norrie disease is an X-linked that results from mutation in NDP gene encoding the Norrin protein, which is involved in vascular development of the retina and inner ear. Mutations in this gene cause Norrin protein being produced with subsequently lack of Norrin protein, which leads to abnormal development of blood vessels. As a result, the retina does not function and ultimately detaches. Children with Norrie disease are born with severe visual impairment. Most children are able to detect light at best, only able to detect light at best. Pupil may appear white on photographs or when light is shown in the eye instead of red. So that's again the exon organization, the protein, and you can see the gene is genetically highly heterogeneous. There are a lot of mutations reported in this gene. So over 75 pathogenic genes have been reported in NDP that cause Norrie disease, with the reports of many novel mutations. These diseases range from genomic rearrangement to single nucleotide mutations, missense mutations, nonsense, and frameshift mutations. As I said, besides the X-linked recessive, there is dominant transfers as well. Dominant inheritance patterns also exist. And X-linked disease, dominant disease is rare, but identifiable by pedigree analysis. There are approximately twice as many affected females as males because females carry a large percentage of X chromosomes. So what you mean by X-linked dominant here, you can see is like if you have only, the women will have one of the X chromosomes, she will still be affected. And the male genotype often in these cases is lethal, but it is viable, if viable, affected males transmit the trait to all their females and none to the male offsprings. So as I said, like X chromosome is not transferred from a father, so it's only from the female, from the mother. And affected females have 50% probability of passing the disease allele to both their male and female offspring. This inheritance pattern may allow localization of disease gene to X chromosome and are important for genetic counseling. So in this example here, the father is having a dominant X condition, and here you can see he is passing this condition to the from the father is affected, is passing to the only to the females and not to the male child. And female can pass the affected gene to both the daughter and their son as well. Coming next is the mitochondrial inheritance. Mitochondria have their own DNA, which is circular in structure, and this is the circular structured DNA of mitochondrial DNA, which actually codes for 37 genes. Genetic alterations in mitochondrial DNA can occur in individual genes, or they occur as larger DNA rearrangements. These genetic changes result in cells having a mixture of normal and altered mitochondrial DNA, which is referred to as heteroplasmy. So this is how they look like normally. This is a cartoon of a normal homoplasmy, where you have this nuclear and the mitochondrial DNA, which are all normal. So you can see this mutated mitochondria is increasing in number. So that is referred to as heteroplasmy stage, and this is the homoplasmy. So how does mitochondrial inheritance takes place? So mitochondrial genes, as you may all be aware, are inherited only from the mother. All offsprings of an affected mother are at risk of inheriting the condition. As you can see in this cartoon over here, the mother is affected and she can pass this thing because the mitochondrial genes only going to be transferred from the mother. There are 100% chances of all the child having the condition. An affected male does not pass his mitochondria, so of course he cannot pass. Even if he is having this condition, he will not be able to pass the condition to the offspring. The most important example of mitochondrial inheritance is LHON, which is labor hereditary optic neuropathy, which is the most common mitochondrial disorder, and it typically affects young males. LHON is a unilateral progressive optic neuropathy with sequential involvement of fellow eyes in the ears, leading to severe bilateral visual loss, was then 2200. Mitochondrial mutations can cause defect in several of the NADH, ubiquinone, oxidoreductase genes chains. This potentially impairs the glutamate transport and increase ROS productions. That's the typical role of mitochondria. So this leads to retinal ganglion cell damage through apoptosis, atrophy, and demyelination, as subsequently noted in optic nerve, chiasm, and tracts. So these are the mutations that are commonly reported in the mitochondrial genome, and LH1 is classically associated with these three mutations, causing amino acid, all causing arginine to histidine and methionine to valine, and all these changes, amino acid changes. They're basically missense mutations. And these three of this, they primarily affect the ND1, ND4, and ND6 genes of the mitochondria. Now we'll move on to some polygenic genes and some multiple inheritance pattern. And IRDs, inherited retinal dystrophy, is a very good example in that, which encompasses a diverse group of genetic eye disease that gradually damage the retina and lead to vision loss. They are highly genetically as well as phenotypically heterogeneous, exhibiting substantial differences worldwide. There are over 300 genes that have been identified in inherited retinal diseases. That's a database. You can have a look at RetNet. And these IRDs can be inherited by dominant, recessive, and X-linked forms as well. So there are two main categories of IRDs. One is the RCD, which is the road corn dystrophy, and the second one is corn road dystrophies, which with each group being characterized by the primary loss of one specific photoreceptor cell, be it rods and cones, and a secondary loss of other cells. So what I mean is, here the rod is lost first and then subsequently cone. In this case, the cones are lost first and subsequently the rods. An example of RCD includes RP, retinitis pigmentosa, LCA. An example of CRD is Stargardt's disease. Congenital retinal pigment epithelium dysfunction is a major cause of both RCD and CRD, and ultimate outcome is blindness. So these are some of the examples of disease and the inheritance pattern. You can see RP and Stargardt, they can show autosomal dominant effect. These are some of the genes. The genes is just an example. Okay, the gene list is very long. So as I said, there are over 300, and RP is also very complex. But just to show some examples, I've put them over here. Recessive, LCA is mostly by recessive and Usher syndrome. X-linked is retinogenesis, which is by X-linked disorder. Now, I just came across a recent review it's basically a review. It's a compilation of all the studies that are done in the Arabic countries. It's a very recent review published in BMC Genomics in 2023. So this study reviewed 1,621 affected individuals from 16 Arabic countries, reported 198 articles. 47 of them were from Saudi Arabia, and that's the whole pie chart showing what all disease they included, with the different types of IRDs that were included. The analysis of the IRD conditions showed that RCD, LCA, and Usher syndrome were widely distributed. If you can see this chart, this is RCD, this is LCA, and this is the Usher syndrome, which are the major conditions that are present in these countries. So what they noted was both RCD and LCA. This is RCD and LCA are the most prevalent non-syndromic form of IRD. On the other hand, Usher, BBS, and Joubert syndromes were the most common syndromic conditions. So this is the pie chart showing for presence of Usher and BBS and Joubert syndrome. So this slide is very busy, but I just included to show what the King's Saudi Arabia studies. As you can again note over here, RCD and the LCA and Usher syndrome and BBS were highly prevalent in the kingdom as well. This is the figure which shows the distribution of the genes that are mutated in these IRDs. Most commonly, they found was TILP1, APCA4, RP1, CRIB1, MyoC, and all these names what I've given over here. And interestingly, all except CRB1 were highly prevalent because they harbored founder mutations, implying that consanguinity is a major determinant in Arab countries. When they analyzed separately for Kingdom of Saudi Arabia genes, they found that TILP1, RP1, and LMS1 were the most frequently mutated genes. So to enhance, as I said, the IRDs are very complex, so to enhance genetic disease management, a decision tree algorithm may be useful in clinical practice. This is actually the case for any genetic disorder, by the way. So what it shows here is you can have a clinical evaluation of the suspected cases. With no symptoms can go for routine examination. With symptoms, they can go for further evaluation. You can refer it to a retina specialist, which can support the diagnosis. Then you refer it to a genetic professional who can recommend genetic testing. If the patient agrees, you can go for genetic testing. If it doesn't agree, then you can go for the routine follow-ups and clinical management. If the patient agrees for genetic testing, you can have the genetic testing done, and depending the outcome, you can subsequently follow up the patients. Oops, sorry. So such an approach can actually help to determine indications for molecular diagnosis and facilitate definitive diagnosis in individuals with IRDs or inherited ocular eye diseases. Now, a second example of polygenic and multiple inheritance pattern is glaucoma. Glaucoma is a complex disorder which includes neurodegeneration of the optic nerve as a defining clinical feature. And primary adult onset glaucomas like open angle closure, pseudoexfoliation are complex, and they rarely follow Mendelian inheritance, simple Mendelian inheritance. But there are other forms of congenital or early onset glaucoma that follow a recessive or a dominant pattern of inheritance. So what this graph shows here, what the cartoon shows here, is that when rare mutations are actually rare and their effect size is very high. But when you look at polymorphisms are very common, but their effect size is very small. So glaucoma is one such example. The adult onset glaucomas is one such examples where where polymorphisms are very high. So common variants actually, they contribute to adult onset glaucoma, but they have very small effect and are potential risk factors. So using this fact, the investigators have actually searched for looking for genes and their associations with disease, different types of glaucoma and their phenotypes. Association studies using candidate gene approach and genome-wide association studies have been particularly useful to identify genetic factors associated with the traits, that is presence or absence of the condition. Identification of susceptible variants have provided novel insights and uncovered new sets of genes. As an example, now this pie chart shows the distribution of genes that were found to be associated with normal tension glaucoma, high tension glaucoma, and the disease itself. There are three big groups been defined by associated with normal tension, high tension, and POG in general. But again, this is just because POG is more than 100 genes have been reported in POG so far. These are just some examples that I'm just trying to show over here, indicating that there is very genetic heterogeneity in these diseases. Now, these are some of the examples where the genes were associated with optic nerve parameters like cup to disc ratio, cup area, and disc area. There were a number of genes that were associated. Total is 38 loci were identified, and among them, they were found 3 genes which were common to all these three phenotypes: ATUH7, SAL1, and TMTC2. Besides that, there have also been genes identified with other traits such as IOP and CCT. These are some of the loci that were identified with IOP, and this is with CAT, CCT, 7 genes in IOP and 26 in this study. Some of these genes were also found to be associated with the POG itself. So 26 loci were identified with CCT, and again there was one common locus, FNDC3B, which plays a role in TGF beta signaling, which was common to both IOP and CCT. As I said, the list of candidate genes for POG is very long. This is just some examples, and whatever is shown in green was what we have actually looked in our studies among the Saudi patients, and with some reporting positive and some reporting negative association with the disease. Same approach of genome-wide studies have been used for angle closure glaucoma, and they have identified so far 8 loci to have a role in PACG development. These are the name of the genes that were identified. And additionally, using candidate gene approach, number of genes have been identified like GF, MMP9, heat shock proteins. And besides, certain candidate genes like SMOC2 and ACVR1 have been actually promising in animal studies as well. Again, studies have identified ACD is a very important risk factor for angle closure glaucoma, and a gene named ABCC5 was also identified to be linked with this phenotype. So this is a pathway enrichment analysis. That's like all the genes when they are looked together in pathways, and what these bubbles you see is the interaction among them, and the darkest pink color shows that they were highly significant. So this pathway gene analysis shows that I don't know if you can see that. This is an extracellular matrix organization, which shows that ECM remodeling may be a very critical mechanism for PACG. Likewise, pseudoexfoliation also has been gene has been identified, which is LOX-N1, and this is the most commonly identified gene in the genome-wide association study. LOX-N1 is involved in collagen cross-linking of extracellular matrix, and has been strongly associated in number of ethnic populations in pseudoexfoliation. And but the only drawback here is this: although they are associated significantly with pseudoexfoliation, the exact role is still unclear, and the presence of this variance in up to 80%, 60 to 80% of unaffected individuals, actually the value of genetic testing is therefore limited and problematic. So as I said, the genome-wide approach, what is the use of identifying all these polymorphisms in a complex trace like glaucoma? There is something like polygenic risk score. It's a way of creating an algorithm to actually calculate the risk. So what does PR examines? What PR does is it examines the cumulative effect of all these genetic variants or traits by aggregating an individual genetic effect into a single measure. They provide a convenient way to quantify the overall genetic risk. So PRS for endophenotypes such as IOP can also be performed in the same way. And in clinical context, PRS can be used for risk prediction in conjunction with traditional risk factors. This is an example of what PRS looks like. You have this case control studies, you do the genome-wide study, you identify the genes, and then later on you stratify, you list all the genetic variants of this patient and add up the risk factors. It's a cumulative index of all the risk factors that you find with significant G, and then you stratify. So your patient will look something like that. The cohort will look like that, and you might identify individuals who are at the highest risk and the lowest risk. So that's just the way PRS can be used subsequently. So it will help stratification, early screening, and maybe implementation of preventative measures. As I said, besides the non-Mendelian inheritance, glaucoma also shows a Mendelian pattern, which a more common example is primary congenital glaucoma. It is inherited in recessive form, and it is very common in countries where consanguinity is common, such as Saudi Arabia and gypsy population of Slovakia. And it is caused by a biallelic pathogenic, since it's recessive, it has to be a biallelic variant in CYP1B1 on LTBP2 gene. Besides that, there has been a TEC gene which is also shown a dominant inherited pattern of PACG. And CYP1B1 accounts for actually up from 75 to 96% of bilateral PCG among Saudi Arabia. Besides that, you have early onset. This is like less than 40 age group, not infancy, but from 3 to 40 years, they are described as early onset glaucoma. These are the genes, MyoC, CYP1, but if you can see, most of them are still unidentified. 80% of patients with early onset glaucoma still do not have mutations in known gene. And clinically speaking, the CYP1 mutation, CCG, which actually has an autosomal recessive, whereas MyoC, Fox, and all the other ones, they are autosomal dominant form of glaucoma, they are clinically indistinguishable. And only molecular diagnosis can actually allow to identify their inheritance pattern and making informed genetic counseling and risk assessment possible. So what's available for genetic testing for glaucoma? Genetic testing for adult onset glaucoma is very challenging. Direct functional evidence of most of the genes that I mentioned is largely missing. Individual variants, they, as I said earlier, only they contribute to small effect of disease risk, indicating that sensitivity and specificity is not sufficient clinically. Identification of epistatic interactions, that is interaction between gene and gene environment, development of PRS or ethnic specific genome panel is needed to identify gene-based genetic testing for adult onset glaucoma. Time being, glaucoma testing seems to be most beneficial for patients with congenital or early onset glaucoma. Coming to the last part of this is what are the diagnosis and management options available. As I said, with advances in next generation sequencing, genomic panel testing has greatly facilitated this diagnosis. So genome panel testing analyzes a panel of genes. So as the name suggests, you only have a specific panel of genes. So there are a number of studies done by NGS this far, for different, even in glaucoma. I'm just quoting certain examples over here. So this is a study where they created their own panel. This is genetic eye disease. They have over 300 genes. So the panel will look like this something. It's like the genes what you're targeting. So using their own panel, they were able to identify mutations correctly in 17 of 18 patients with known IRD, glaucoma, and optic atrophy variants. The sensitivity was quite good. They identified 51% cases where the clinical phenotype and genotype are very good correlation. The reproducibility of this panel testing was very high. And the study suggested that panel testing offers a higher sensitivity, specificity, and quicker turnaround time. So just to say what this highlight is, you have a genome panel testing as an option for doing your genetic testing. The second approach can be, you must have heard, whole exome sequencing. So whole exome sequencing is a powerful technique that is used for genetic testing that focuses on sequencing all the exons throughout the genome, which actually make up less than 2% of the genome and contain over 85% of the disease-causing variants. Again, there was a study recently where they did whole exome sequencing in 90 patients with suspected inherited ocular diseases. These are the different types of disease that were included in the study. And again, with this, whole exome could provide a definitive diagnosis by 58% of the probands. And these are the various genes. As I said, all these types of genes are very heterogeneous. So you can see the number of genes that were identified in all these disorders. But again, whole exome is also one of the approach. Next is the whole genome sequencing. As the name suggests, whole genome, unlike panel or whole exomes, targets the entire genome. It provides a comprehensive information about all genes. It's an option where targeted or a whole exome fails to identify genetic diagnosis. Allows identification of wider genetic variants, upstream, regulatory, non-coding, coding, copy number, and structural variants. Again, there was this recent study done in UK where over 1,000 families underwent genetic testing for posterior segment, anterior segment, ocular malformation, and albinism. And this study was able to diagnose 55% of the cases. And in this 183, the diagnosis was missed, and around 28% of this patients were actually previously tested to be negative by targeted testing, highlighting what are the advantages of how it can help you identify more patients. Now, the treatment options available so far for for genetic disease is only gene therapy, which is RP65. Everybody must be aware of this. Patients with this biallelic RP65 gene defect, they actually lack enzyme cis-retinal essential for 11 cis-retinal, which is essential for phototransduction, due to absence or non-functional protein causing photoreceptor degeneration. So the therapy actually aims to halt the retinal degeneration by injecting a healthy copy of RP65 in adenovirus by a subretinal injection, and it enables to synthesize RP65, normal RP65. With this functional RP65, the visual cycle is restored. So that's a gene therapy which is available. And besides this RP65 mediated gene therapy, which is very revolutionary, it comes with a cost and cost benefit analysis and has yielded varying results. There are additional therapies of gene editing, optogenetics, stem cell therapy, and retinal prosthetics, and neuroprotective therapies such as N-acetyl, cysteine, ciliary neurotropic factors, rod-derived cone-viable factors. So basically there are other therapies as well in investigation. So just to summarize, what's the role of all the genetics in clinical practice? It can provide precision diagnosis, targeted genetic testing, analyzing specific gene-associated suspected eye disease, allow more accurate diagnosis, especially for complex diseases which are challenging, clarifying phenotype spectrum. Genetic testing can differentiate between similar presentations, guiding appropriate treatment and prognosis. And predictive testing, identifying genetic carriers, inform family planning and early intervention for potential risk in offspring. It can help facilitate personalized medicine, genotype-guided therapies, understanding the genetic basis. That example of RP65 is there. Then emerging gene therapies, as I said, are also going on that have a whole promise. Then it can allow risk stratification. Genetic markers can identify individuals at high risk for developing specific eye diseases, enabling monitoring and preventive measures. Besides that, they can provide enhanced clinical decision making. Genetic counseling, collaborating with genetic counselors, empowers patients and families to understand their genetic risk and make informed decisions about their health and potential treatment or management modalities that can be available to them. It provides improved prognostics. Genetic information can refine our own understanding of disease course and complications and facilitate treatment. Clinical research, integrating genetics into clinical research studies will allow rapid discovery of new diagnostic tools, therapeutics, and maybe even personalized strategies. What are the challenges? Investigating genetic landscape in Arab world. While some genomic studies have been reported in Saudi Arabia and the broader Arab population, but large genomic population-based studies are still less. Establishing population-specific biobanks or cohorts tailored to Saudi population, or fostering international collaborations and with using cutting-edge technologies can unlock wide spectrum of data and advance our understanding of eye disease in the region. And although whatever I mentioned, those panel testing, exome, genomes, or whole genome serve the purpose for molecular diagnosis, despite these genomic advances, half of patients, they remain undiagnosed. You must have noticed there are 52%, 58% even on whole genome sequencing. So there are still half the population which is not unable to diagnose by using these methods. And the proportion is much higher in individuals with unilateral or even milder phenotypes. Furthermore, even when a strong candidate gene is available for patients, issues of incomplete penetrance and mosaicism makes diagnosis and genetic counseling challenging. So this part actually highlights the importance of clinical phenotyping of patients. So prioritizing research in the region in eye disease will help inform public health by informing prevention strategies and personalized treatment. Clinical diagnosis and management are guided by phenotypes. So we have to remember that clinical diagnosis and management are still guided by phenotypes and not genotypes. However, although genetic testing is not needed to manage treatment, it can be instrumental in specific clinical scenarios. Thus, integrating ophthalmic genetics into clinical practice will empower clinicians with molecular diagnostics options when needed and enhance decisions for dealing with patients with genetic eye disease. This can potentially lead to improved vision outcome and quality of life. Thank you very much.
Thank you very much, Dr. Altaf, for this nice presentation. Congratulate you for simplicity of giving this inheritance mode of genetics in a simple way, if you agree with me. Now we'll open the floor for any questions, please. If no more question, I can اتفضل دكتور حسين.
Yes.
Thank you very much for excellent presentation. Very important subject. Actually, we have many congenital diseases here in the kingdom as well as worldwide. very important because we have so many patients, Dr. Kadan and others, Dr. Hassan, Dr. Masfir, they know more about it. So I think it's important to include it. Unfortunately, most of the genetic disease, which will take place after having the disease.
Yes.
And when the patient suffer, it's a matter of just only just
Pre-test.
record and case reported and these things. So we need to convert this to a useful data, and especially for congenital glaucoma because we have so many patients and retinitis pigmentosa. Thank you very much.
I agree with you. So what I understand you are trying to say is like congenital glaucoma testing should be done before. I think that's what I think people do normally. People, when they are aware if they have a congenital glaucoma, they go for testing like to follow up whether they have these common mutations or not. Yeah, you mean to say before getting into the stuff.
It's not one of the pre-marriage tests.
I am sure, sorry, I don't know on that if there is actually a premarital test panel includes congenital glaucoma. But you are right, I agree, it should be included, considering that consanguinating among this population is very high.
Dr. Layla?
We actually started in KKISH implementing genetic counseling for families with congenital glaucoma. And we found out that 80-something% of them are related to CYP1 mutation, the autosomal recessive, which if we stop, we'll stop 80-something% of future generation. But still there is something about our patients, like they don't like to tell the bigger family. They only tell the core family about the genetic testing and the result. But we are still educating them, pushing them. And we have done almost 400 something tests here in KKISH for index patients, like one patient from family, no matter how many affected. And they are bringing their siblings, many families we referred for, we found the gene mutation and we referred them for a test tube baby with the pre-implantation genetic testing, for further, to get further kids in the family not affected by congenital glaucoma. So hopefully in the next few years or tens of years, we can eradicate 80-something% of congenital glaucoma in Saudi Arabia.
According, he is actually want to be done before, not after we detect the patient. Yes, yes.
So they can include
exactly.
pre-marriage
But if you calculate still to do for the whole population, it's only the affected children in 1 in 3,000 to 10,000. And many of congenital glaucoma are living very normal life if they are detected very early, treated very good. So it's not that much of disease. It's not a killing disease or not. And still families are, I think the premarital testing, as I know, it's not genetic testing. It's something chemical test for the sicklers and thalassemia.
Yeah, they normally, I don't know for
It's
which
not
is
genetic,
available in this kingdom, but it's done for
Yeah, I've
thalassemia
heard
is a very
it's not-
common one. Yes.
Yeah, it's not a genetic
Yeah,
testing.
it's not a genetic
so,
testing.
genetic testing takes a while. It's available actually here in the labs between 400 to 2,000 according to what you ask for a single gene or a whole genome sequencing in the private, and they do even siblings for free. So, but the main problem is educating the community about the mode of transmission and that we can stop the disease.
So this will lead us to, has questions? Dr. Mesfeh?
I appreciate you bringing up the point of, sorry, I wanted to say something.
Thank you very much, Altaf. It is a very interesting subject for all ophthalmologists and all the medical field practitioners. I'm very optimistic, and I think currently we have different members who are interested to do genetic study, genetic research, genetics investigation to get a novel mutation. But I think, to the best of my knowledge, in UK, they are doing some genetic testing for oncogenes, where they could predict those patients who may have at high risk to have oncology situation diseases. I think, according to my knowledge, the Ministry of Health, I think they allocated some lump of money for preventive measure. I think it's not only congenital glaucoma. There are many diseases could be prevented, including wide range of hereditary diseases that maybe either could be oncology, could be oncology cases, could be non-oncology cases. How could we, as a developing country, establish such system?
I think that's a very policy decision-making kind of information, you know, like how in the kingdom by collaborations, by awareness and things like that. It's a what you are talking about is a very large-scale kind of approach, you know, and not to a specific genetic disease itself. You are talking about all the genetic diseases. So
when they did the Saudi genome, do they go through these things? What is the exact mission to know the Saudi genome? What they will obtain?
I don't know if they're actually looking at analyzing these genomes to see what the inherited pattern looks like. I do not know on that. So
No, what is the purpose of doing Saudi genome at the end?
maybe they are mapping all the information what the genetics looks like in the population. I don't know if they are talking about only the healthy people or even the diseased one. So that's a different thing.
Actually, we look for two things: prevention of the disease, how to stop, and second thing, if there is way of treatment to get. But this is the main aim of looking for these things. Yeah.
I would like to go with preventive measure rather than gene therapy.
Yeah.
Gene therapy is
Still poor.
costly, still premature, still investigation, but preventive measure is more accurate, more preventive, more successful, more rewarding.
Because if you use genetic testing for your information or data sake, you will ultimately have to find a solution which is gene based. You know,
This
what
is
I'm
need
trying
a
to say is
scientific research, high advanced lab, not. But
Yeah.
preventive measure, I don't think it will need that.
Preventive measures, I don't know, to handle gene defects can be done by preventive measures. Like, you know, maybe for a CYP1B1, as we're talking, consanguinity is the main problem. So if we identify that, we stop the marriages. He was talking about premarital kind of testing, you know. So that can be a preventive measures. But
I think if we have a project to do preventive measure, after maybe a few decades, we'll just end it with the new sporadic cases.
Yes.
that's possible.
Anyway, Dr. Altaf, for congenital glaucoma, as we most of talk about congenital glaucoma, sometimes we receive very simple cases in which any type of surgery you do, the patient will control all his life. And sometimes we receive very severe cases of coronary scar, multiple surgery, and the failure is very high. My question, this is about phenotype and genotype. Does the inheritance will affect the severity of the disease, whether it is autosomal dominant, autosomal recessive? And because of congenital glaucoma in Europe, we never see these cases, what we are seeing here. How is your explanation?
See, I think those genotype-phenotype correlations, there are not many studies which are actually looked at in very details. You know, as you said, some phenotypes are very milder cases, but still they might have the defect. So that it is because some genes, they are not expressed entirely. Like for CYP1B1 is a recessive, where it requires both of them to be abnormal. So even if you have, you can see clinical variations. It because how much of protein is expressed. So that expressivity part is a totally different story.
This is very important point. If we look for this, we may have mild cases of congenital glaucoma.
Sorry, I studied a lot about the CYP1B1 mutation. Actually, in Europe and America, they found that CYP1B1 mutation was associated with the worst clinical phenotype. So, and a high failure of surgeries. And even with the juvenile open-angle glaucoma here in Saudi, we published something, KCASH, with King Saudi University. Also, CYP1B1 mutation was 80-something% responsible of the glaucoma. And we found also homozygous CYP1B1 mutation that are normal. It could be or a little bit bigger disc or a little bit bigger eye, but they didn't express the whole disease. And there were studies of about gene regulating proteins or something, but they came all negative. Okay. So I think till now we have only to prevent the pre-marriage counseling and testing. This is the only thing that's available.
Thank you. Any more questions?
About these tests, like the 23andMe, are they good, like for diseases and?
I'm not aware of that. How
Okay.
good they can be, like for
Okay.
23andMe. It's such a huge project they keep doing. Yeah.
Thank you very much. If there is no more question, thanks, Dr. Altaf.
Thank you
And
very
we can
much.
start our quiz, please.